Hello, everyone, and welcome to the Global IMMUNOTALK. It is a great pleasure today to introduce Dr. Lai Guan as today's Global Immuno speaker. Lai Guan is originally from Malaysia, and he completed his PhD work at the Garvan Institute of Medical Research in Sydney, Australia in 2004. He then joined the Institute of Molecular and Cell Biology in Singapore for his postdoctoral training under the supervision of Professor Lam Kong Peng. In 2006, Lai Guan joined Wolfgang Weniger's laboratory at the Wistar Institute in Philadelphia. And this is where he started with his research interest in intravital microscopy, techniques to study leukocyte biology. Following his postdoctoral training, he joined the Singapore Immunology Network to establish his own laboratory in 2009. In Singapore, he is now adjunct associate professor at the National University of Singapore and also an adjunct investigator at the National Cancer Center in Singapore. Lai Guan's primary research focus is the study of myeloid heterogeneity, myeloid cell heterogeneity, particularly neutrophils. He very cleverly uses a combination of in vivo imaging models. I'm sure we will see a lot of that today. Mass cytometry, flow cytometry, transcriptomic and bioinformatics approaches to determine the developmental pathway of leukocytes, as well as the cellular and molecular events involved in the regulation of leukocyte homeostasis, and particularly neutrophils. Lai Guan has been recently transitioning to a new institute in Shanghai, China, the Shanghai Immune Therapy Institute, where he's connecting from today. So Lai Guan, thank you very much for having accepted our invitation, and we very much look forward to your talk entitled New Concepts for Good Old Neutrophils. Now, before you start, it is a tradition of the Global IMMUNOTALKs to ask speakers a question to get to know them a little better and to perhaps inspire the next generation of scientists. So the question we have chosen for you, Lai Guan, is could you share one of the most impactful decisions in your scientific career?
Sure. So first of all, thank you, Matteo, for the kind introductions, and also thanks all the organizers for this opportunity. And to answer your questions, I think the most impactful decision I made is actually 13 years ago. I moved from Sydney to Singapore to set up my own lab, even though at that time my former supervisor, Wolfgang, is kind of creating a small group for myself, but I decided to actually move to Singapore to a very new institute at that time. And I think that actually changed my career quite a bit, also my life in many ways, because I get to know many people. For example, like yourself, and also lots of good colleagues within the institute, and also in Singapore and abroad. I think Singapore being a center for transit, so we get to meet a lot of people transiting through Singapore. So I think that was a really impactful decision that I made 13 years ago. So I hope in few years' time I can also tell you that another decision that moving to Shanghai will pan out well, and then I will look back and say that, oh, it's another impactful decision that I made recently after 13 years in Singapore. Yeah,
I very much hope so. Thank you, Lai Guan. You can go ahead and share your screen.
Sure. Okay. All right. So you can see on my screen.
Perfect.
Perfect. Okay, cool. So I'll get started. So I would say that good morning, good afternoon, and good evening to all of you coming from different parts of the world. For myself, it's about 12:04 a.m. So today I'm going to go through some of the projects that we have been doing in the lab in the past few years. Some are published, and then the second part of it will be unpublished data. So let's just get my monitor. So as you may know that our neutrophil are made out of 60 to 70% of the circulating leukocyte in human, and it's about 20 to 30% in mice. And they play a major role in defense against microbial pathogens. And they are amongst the first blood-borne cell to be recruited into inflamed and infected site. So I just have to close the chat. Okay, sorry about that. And because of they are the first cell to be recruited to the inflamed or infected site, how they carry out their function is to release mediators and enzyme that can get rid of the invading pathogens. And however, this can also damage the surrounding healthy tissue. Because of that reason, the production, release, migrations, and clearance are tightly regulated. So as Matthew mentioned before, in my lab, mainly when we started, I was kind of we do a lot of imaging as well. So this is just to show some of the imaging technology or platforms that we set up in our lab. So one of it will be like a 3D imaging, bone marrow imaging, in which we can optically clear the bone, and this will then allow us to visualize the neutrophil within the bone marrow. Without sectioning, without doing the sectioning, and then we'll get a 3D context within the bone marrow to visualize the neutrophil in a 3D context. And more importantly, we also do a lot of dynamic imaging using a 2-photon intravital imaging. And as you can see from this first movie that I'm showing here, when we injected GCSF, which is a neutrophil mobilizer, you can see that how this neutrophil kind of moving from this parenchyma into the sinusoid and being released into the circulations. And if you move to this particular movie, I'm showing that how neutrophil respond to an injury in the mouse ear skin, and we define this recruitment as a 3-step cascade, in which it start off with scouting, whereby firstly a few neutrophil will get to the injury site, and this will then trigger amplifications phase, whereby a lot of neutrophil will be recruited. And this will then form a stabilized cluster around this injury site. And this movie, you can see that like we are looking at how these neutrophil respond to immune complex formations within the blood vessels. And these are the immune complexes forming deposited on the blood vessels, and how these neutrophil kind of adhere to the blood vessels. And when they transmigrating out, they can also carry some of these immune complexes with them. And one of the reasons why I'm showing all this movie is just to illustrate that how neutrophil actually work. They don't work like 1 cell or 2 cells. They work in a large numbers. And the other features of their function, their function is that is through the rapid mobilizations. However, this will then pose a problem for our immune system, because as you may know that neutrophils are relatively short-lived. They have a half-life of 6 to 12 hours in the circulations. And because of this reason, to meet the demands, neutrophil production actually represent the majority activities for the bone marrow. In fact, about 2/3 of this hematopoiesis is devoted for myelopoiesis. And because of this reason, our lab started to become interested into studying neutrophil development in the past few years. And one of the things that we realized at that time, classically, how the field or hematology field characterized neutrophil developments is through these methods by cytospin, in which they will look at the granules and cytosol content, granules contents within the cell through the cytospin, and this cell can then be characterized into myeloblasts, promyelocyte, myelocyte, metamyelocyte, and band cell. And however, this kind of a characterizations method it doesn't actually allow for any downstream analysis or molecular analysis. So because of this reason, we then started off to think about whether can we find a way to better characterize the entire neutrophil developmental pathway. So the first thing we did is to, as you may know, that all the progenitors or precursors of leukocyte, they are highly proliferative or they are proliferative. The reason being is that they have to proliferate to give rise to enough number to make up enough number for this leukocyte. So because of that reason, what we did in 2018 it was that we used this cytoph together with 40 different antibody together with the IDU, which will then tag the proliferating cell. And based on this approach, we can then look at a wide range of differentially expressed surface marker between the proliferative and non-proliferative neutrophil. And based on this, because this data is already published, I won't go into details. Essentially, what we observe here is that we identify a population of cell in the bone marrow in which we name pre-new neutrophil precursor, which are proliferative, and they are CXCR4 high and c-KIT high. And within these CXCR4 and c-KIT low populations, when you get it, essentially you can then further separate them into mature neutrophil, which is resembling those that in the blood, and the CXCR2 low or negative immature neutrophils. So based on this characterization, we then can start. We then define the developmental trajectory from the GMP to the pre-new to immature neutrophil in the bone marrow, and this mature neutrophil will then release into the blood. After the paper published, Emmanuel then actually came up with these questions. So when he cultured the GMP or the pre-new, which is like proliferative, as I mentioned before, however, he realized that this pre-new only can divide 2 or 3 times, whereas this GMP can form these colonies, but not for the pre-new. Where and immature and mature neutrophil, they are post-mitotic cells, so they can't divide any further. So which also means that if pre-new do not actually form colonies, how would this precursor cell give rise to enough neutrophil? So this doesn't really make sense to us at that time. So we then start to ask the question, could it be that how we actually define the GMP, which is the granulocyte monocyte progenitors, how we define this cell or these populations of cell may actually consist of a committed neutrophil progenitors that we still haven't defined. So because of that, we then started to do more experiment. First thing that we did, Emmanuel did, was to extract the GMP data from this paper published by Ido Amit's lab. So essentially, he extracted the GMP data from their single cell data set. And from here, he performed clustering. And as you can see here, by doing these simple clustering plots, you can see that GMP can be clustered into 10 different populations, which include monocytic populations, basophilic, eosinophilic, and also a neutrophilic population here. So, which is good. It shows that GMP can actually contain a mix of committed progenitors. But then the question will be, how can we isolate these subpopulation for downstream analysis and validations? So that would be the questions. So to do that, we make use of this infinity flow workflow that established by Evan Nguyen's lab in SIGN at that time. And essentially what we did here is isolated the bone marrow cell from the mice, and we stained them with the 13 backbone markers, in our case as neutrophil markers, and we leave the PE channel blank, and we use a legend screen kit which consists of 255 PE conjugated antibody, and we screen these 255 antibodies by flow cytometry. And from here, with this data set, then we ran through this infinity flow algorithm. And essentially, you can imagine that we take these 255 together with the 13 backbone markers. We kind of compress it into one single FCS file, which is a one flow cytometry analysis file. And from that, we can then analyze all these antibody staining together. So from there, we can then get on the GMP, which is the CD34 high and CD1632 high cell. And again, you can see from this infinity flow data set, we can cluster this cell into 10 different populations. So further validating the transcriptomic data set observations. Then next question will be, how do we find the most useful marker for putative neutrophil progenitors? So since we already know the markers for pre-new and the immature and mature neutrophil, so by using this UMAP plot, which has the advantage of showing the interrelationship and providing more global view about this interrelationship between different cell type. When we plot this, projected this infinity flow data set to the UMAP, what we can see is that how this GMP is kind of connected to the neutrophil lineage and monocyte lineage. And for us, we are interested to find the putative markers that can help us to identify the progenitors, neutrophil progenitors. So in that case, we then want to figure out like what will be the markers that is kind of bridging the GMP to the pre-new, which is this region here. So to cut the long story short, by doing this kind of screening, we then identify several markers. And based on these markers, we can then further split the GMP into Ly6C high GMP, Ly6C low GMP. And from this Ly6C high GMP, when we get on it, we can further split them into the pro-new, which is CD81 positive, and then the CMOP, which is the common monocyte progenitors. So in other words, this pro-new will probably represent the in the same ontogenic hierarchy as the CMOP for the neutrophil in that sense. And within this pro-new, we can further split them into pro-new 1 and 2 by CD106. With this marker, having identified this marker to kind of subsetting all the neutrophil subset within the bone marrow, we then sorted them and perform RNA-seq. I should say that perform the nanostring and bulk RNA-seq. Sorry, bulk RNA-seq. And what you can see here is that the Ly6C negative GMP is kind of in the middle, which is kind of nicely that split into this neutrophil lineage, pro-new 1, pro-new 2, and pre-new, and also the CMOP and then TPMO, which is the monocyte lineage. And more importantly, when we look at the monocyte transcription factors amongst all these subset that we identify, you can see that for those monocyte transcription factors, it is upregulated in CMOP, whereas it's kind of downregulated as the cells committing towards neutrophil lineage. And these are including the RRF8, KLF4. As you can see, this is downregulated in pro-new 2. Conversely, for the neutrophil transcription factor, for instance, the GF1, CBP epsilon, and PER3 is upregulated as the cell committing towards the neutrophil lineage and downregulated in CMOP. So it kind of suggesting that those subset that, or I should say that those markers that we identify should is helping us to subset these various progenitors and precursors nicely. And this is just kind of a heat map to summarize what I've been mentioning. You can see that within this CMOB clustering, you can see all these monocyte genes are highly enriched, whereas for the Ly6C is almost like haven't decided whether they are going to the neutrophil lineage or the monocyte lineage, because they are still expressing both neutrophil transcription factors or monocyte transcription factors. Whereas for the ProNeu1 and ProNeu2, you can see that it's highly enriched in all these neutrophil related transcription factors. And if you take if you look at this CBP epsilon, it has been well known that CBP epsilon knockout mouse, they do not give rise to mature neutrophil. However, this developmental stop is unknown because previously there is no good way of subsetting these different progenitors and precursors within the neutrophil developmental trajectory. So with our way of identifying each of these subset, we then look at both the wild type mouse and the CBP epsilon mice. You can see that with this wild type, you can get like from the GMP, you can get ProNeu1, 2, pre-new, immature, and mature neutrophil. Whereas for the CBP epsilon, the development arrest actually occur at the point of ProNeu1. And interestingly, once this arrest stop at the ProNeu1 stage, somehow there is an appearance of this atypical granulocyte, which is a F positive and CD11, CD115 positive. So by having this very nice way of looking at the entire developmental trajectory, we can then identify when you do not have this CBP epsilon transcription factors, the arrest actually occur at this stage. So to further prove that those markers that we identify can faithfully help us to sort out these ProNeu1, ProNeu2, and pre-new, what we did here is that we perform a transfer experiment in which we combine, we sorted ProNeu1, ProNeu2, and pre-new in essentially in 3 different colors, if you wish, and combine them in 1-to-1 ratios. And after that, we then transfer them into a wild type recipient mouse. By doing so, we were then able to to study them within the same control environments. And after 3 days of transfer, we then perform an analysis. And as you can see here, the ProNeu1, GFP positive ProNeu1, after 3 days, most of it is still remain as differentiate until the immature neutrophil stage. Whereas the ProNeu2, some of these cells already are differentiating, maturing into the mature neutrophil. Whereas the pre-new, almost 70% of it is already becoming mature neutrophil. So this is just a summary to show that since the ProNeu1 is the so-called the earliest progenitors, so after 3 days, most of these cells can only differentiate into the immature cell. Whereas the ProNeu2 is 1 step ahead in this maturation step. After 3 days, most of them are still immature, but you can get some mature neutrophil already. Whereas pre-new, most of it will become mature neutrophil. So again, it's just showing that what we sorted is committed progenitors. And one point that I forgot to mention is that more importantly, if you look at this gate, which is the eosinophil or monocyte gate, even after 3 days of transfer, you don't get any of these eosinophil or monocyte. So it kind of further show that the ProNeu1, ProNeu2, these are committed neutrophil progenitors. So we in 2019 with Renato and Andreas Hidalgo, we actually wrote this perspective in Nature Review Immunology, in which we talk about perhaps there is a existence of a pro-neutrophil before pre-new, which is kind of fortunate for us that we show in 2020 that ProNeu1 is actually part of these GMP populations, and this ProNeu1 can then develop into ProNeu2 and continue on to become mature neutrophil. So I also want to take this opportunity to highlight that in the past few years, there are lots of effort going into trying to somehow unify, or I should say that somehow unify how we should look at neutrophil subset. And this work has been done not only from my own lab, for instance, Hendrik's lab, Lee Grimes, Lo Hongbo's lab, and also Kang's lab in Korea. This lab has also been putting quite a lot of effort into trying to characterize neutrophil, and while we still cannot fully agree on how we should name them, as you can see, the naming could be quite different in different papers, but most of if you really compare them side by side, some of it is just the same cell that we are looking at, but it's just we name it differently. So hopefully there will be a day that we can actually unify all these naming system, and will make it easier for people to understand neutrophil subset. So you may then ask me, how about human? So we did actually use the same approach, legend screen, to screen for different markers for human neutrophil. And I just want to quickly go through this, because this data is already published. So essentially, we use a human corplet and we use a legend screen approach to help us to identify different markers that can stain for pro-neu all the way to mature neutrophil. And this will be the kind of summary plots that you can see that from pro-neu all the way to mature neu, we can identify using different markers. But I have to admit that we just provided very basic information how to stain this neutrophil in human. And more recently, there are multiple very good papers that have looked into this neutrophil development in much more details compared to us. For instance, papers from Hendrik's lab, from Casatella lab, and also more recently from Renato's lab as well. So if you are interested, please also refer to these papers. So now we learn how to kind of subset neutrophil under the resting state, and we thought that perhaps we can use this system and try to understand better tumor neutrophil. Because as you may know that currently how people name neutrophil, tumor neutrophil is purely based on their function. They can be named as N1, N2, MDSC, or tumor-associated neutrophil. So, however, this is unclear whether this neutrophil is actually is emerging from a new subset, or actually is a result of preferentially recruitments of a certain subset of neutrophil to the tumor, or actually this is a tumor-induced state. So these are the unknown questions, and because of this reason, I also think that because of this reason that is tumor neutrophil is still kind of an unknown identity. So to help us to understand our neutrophil heterogeneity in tumor, we made use of this KPC autotopic model, which is the pancreatic tumor models in mice. So what we did here is we transplanted this cell line into pancreas, and we wait for 6 weeks, and then we harvest all these different organs, including the tumors, and we sorted the neutrophils, and we barcoded them, and we ran 10x single cell RNA-seq. And this is the UMAP of when we combine all the data sets from these different organs and including the tumor, and you can see that it gives a very nice view of this neutrophil development continuum from proneo all the way to the mature neutrophil, and also a tumor cluster. But when you break it down into different organs, for instance, like in the bone marrow, you can see that there's this very nice trajectory from prenue all the way to mature neutrophil. And then also in the spleen, you also get some of this, and but most of them are mature neutrophil, same as in the blood. Whereas the tumor, you have this very unique cluster that can be further subdivided into three different subclusters. And when we look at the frequency of the cell, we can see that within the tumor, they are quite unique. So you have these three population here, or I should say three cluster. So after we got this data, we then want to kind of integrate it with a steady state. So we make use of this data set that published in Nature Immunology by Lo Hongbo's lab in 2020. So this is their data set, is indicated in this yellow color. And if we integrate our data or overlay our data on top of it, you can see that essentially there is no deviations in the bone marrow developments, or I should say the neutrophil development trajectory in the bone marrow, even though the mouse are tumor bearing. And the only thing that we did actually observe, I should say two things that we observed, is that this tail that are representing from the prenue, immature and mature neutrophil, so in the spleen, so it kind of indicative of there's this increased EMH, extramedullary hematopoiesis, and also this cluster that is unique to tumor. So it kind of show that so the two things that we observe, EMH and also unique cluster that you can detect in the tumor. So I want to draw your attention to these three sub-cluster, T1, T2, and T3. So the reason why we name it T1, T2, and T3 is that tumor neutrophil 1, 2, and 3. So the first thing we did is that we want to look at their maturation status. So by doing that, we hope that then this will give us some indications where they actually coming from. And you can see that when we do this analysis, what we observe is that the T1, which is this population here, is kind of corresponding more to this immature neutrophil, whereas the T2 represent more of this mature neutrophil, and T3, which is kind of a mix, is kind of in between mature and immature. So this is kind of interesting to us. So when Melissa look at this using the diffusion map, what we could see is that the this kind of it gives kind of indications on how this trajectory works. So it starts from this prenue, and then it go to the immature neutrophil, and then it go to the mature neutrophil. So which is the normal kind of neutrophil developmental trajectory. And what we also observe is that this T1 is kind of coming from this immature neutrophil, and then T2 is kind of arising from this mature neutrophil and converge at this point, forming this T3. So this is kind of interesting. Then it kind of raises the questions that does it mean that both the immature and mature neutrophil can be recruited to the tumor tissue? Indeed, that is what we actually found in our previous paper. If you look at this plot here, in the tumor, we can detect both the mature neutrophil and also immature neutrophil. And just to illustrate that both the immature and mature neutrophil, they have the ability to be recruited to the injury site or to an inflamed site. We perform this intravital imaging by mixing this 50% 1-to-1 ratio mature and immature neutrophil in two different colors, and we then transfer them intradermally, and we induce this laser injury. And you could see that both these cells can still respond to the injury that induced by the laser. And again, this will then kind of confirming that what we observe here, both the mature and immature neutrophil are able to migrate or recruited to the inflamed site. So with that, then this also raised the questions. So if both of them, T1 and T2, can be recruited to the tumor tissue, what does it mean? Does it mean that they can directly become T3, or T1 has to become T2 first and before it can become T3? So these are the questions that we don't really know. And Melissa also performed the RNA velocity analysis, and you can see that essentially what this plot is showing, that T1 and T2 most likely is representing a transitory neutrophil state that will eventually differentiate to become T3. So that was kind of our hypothesis. And to validate that, we then perform a ataxic analysis of immature and mature neutrophil. And how we did that is that we sorted immature and mature neutrophil from the tumor-bearing mouse in different organs, including the tumor, bone marrow, blood, and spleen, and compare this with the wild-type mouse. And when we look at the open chromatin regions for the T3 genes, immediately you can see that only in the tumor, regardless of whether they are immature or mature neutrophil, we then observe the open chromatin regions in these cells within the tumor, but not in blood, spleen, or bone marrow. So it's further suggesting that all these reprogramming most likely happen within the tumor. So this will be what we are so far, all our data is kind of pointing towards. So to kind of explain it in a more easy way, perhaps, like some of you guys may know Foron, so although with the mask, so you can imagine two of us are very different persons. So we are in this different room, and then the moment that we go into the same environment, we then put on the same kind of t-shirts, so doing the same work. So which also means that this will be the reprogramming. So regardless of how mature or immature we are, but the moment that we get into the same environment, we are reprogrammed the same way, we are put on the same shirt, and then doing the same kind of functions. So that is how we see the reprogramming happening within the tumor microenvironment. And when we also look at the GO pathway, we also observe that each of these clusters, they have a very distinct enriched pathway. For instance, the T3 populations, they are enriched in terms of their angiogenesis pathway and glycolytic process pathway, and also a hypoxic pathway. Whereas for the T2 is more like related to effector functions, and T1 is more like an immature kind of a cell type. So having shown that these three populations, they have a very distinct kind of a signature, we then start to ask ourselves, can we actually resolve this in spatially? So before we go there, I just want to quickly go through these two key points. So essentially what we have shown here is that the epigenetic reprogramming towards the T3 fate is detected in both immature and mature tumor infiltrating neutrophil. That is from our ataxic data, we show that. And also we also show that most likely these two, T1 and T2, they will further differentiate and converge and forming the T3 neutrophil. So the next question that we want to ask whether we can spatially resolve all these three populations within the tumor. So to address this question, we then perform the 10x Visium transcriptomic analysis. And how we did that is essentially we cut the tumor into 4 kind of quadrants, and with that we then section it and place it on this Visium slide, and this is a representative tumor sections. And after we perform the analysis, and you can see that in which this is the core, which is lack of any signal, and within this tumor regions, you can see a different cluster. And when we perform this UMAP clustering, you can we could observe that there is a 10 different cluster. And since we already have the signature for T1, T2, and T3 neutrophil, we then take this signature and we project onto our spatial transcriptomic data set. And these are the 4 different sections. And essentially what we observe is that specifically for T3, somehow they are localized around this necrotic region or the tumor core, near to the tumor core. As you can see from this section here, as well as this section here, and also this section here, and also here. And also when you project to the UMAP, you can also see that the T3 distinctively cluster together here, whereas for the T1 and T2, they are more kind of scattered around. So the other question that we also ask, like, where can we actually find this T3 neutrophil? So when we look at these sections specifically look at the hypoxic or glycolytic or angiogenic regions, and when we project the T3 signature onto it, or I should say the T1, T2, and T3 neutrophil onto it, what we found is that essentially, if you look at this graph here, T3 are predominantly located at in this region, but not for the T1 and T2. So it kind of suggesting that perhaps T3 are those cells that are pro-tumoral neutrophil. So I think I mentioned this, so I will skip this slide. So having shown that most likely that this T3 neutrophil is kind of switched to more like a glycolytic and hypoxic profile, which is indicative that they are pro-tumoral. So we ask ourselves that could this be similar kind of reprogramming is happening in a human PDAC? So to address this, Melissa took this data set from these 2 papers from human PDAC patients, and extracted out their neutrophil data set and label transfer to our UMAP, the reference UMAP. And as you can see here, with this PDAC tumor neutrophil, when it kind of label transfer, it kind of fits quite well, it mapped quite well to our tumor cluster, including the T3. For the adjacent normal tissue neutrophil, most of it is just kind of mapped to the mature neutrophil cluster. And then same for the other data set. As you can see, the PDAC, from the PDAC neutrophil, it mapped quite well to our tumor neutrophil cluster, and the peripheral blood only mainly map onto this mature neutrophil cluster here. So having shown that similar reprogramming is occurring in human, we then ask the clinical relevance of T3 neutrophil. So we want to test whether T3 neutrophil signatures can be a predictive kind of predictive markers for pancreatic tumor cancer. So we then kind of we then perform this TCGA analysis. If you look at this graph here, with the high T3 with the high T3 signatures, essentially you have a poorer survival in both cohort that we look at from this database, TCGA database, as well as the Australian cohort. You can see that with the high T3 signatures, you have a poorer outcome. And similarly, when we look at the disease-free survival, when the patient have a high T3 signatures, they have a worse disease-free survival in both these cohort that we look at. So based on this, we then concluded that T3 neutrophil signatures are associated with poorer outcomes in human cancer, pancreatic tumor cancer. So to summarize my talk, essentially what we show here is that in response to the presence of tumor, there's increased generations of neutrophil. However, this increased generation of neutrophil still preserve how the neutrophil mature. The sequence is still preserved. And with that neutrophil, with the increased neutrophil, this neutrophil will then be recruited to the tumor. Both the mature and the immature neutrophil will be recruited to the tumor, and they will undergo differentiation to become the T3. But one of the key points that we observe here is that if you look at this part, definitely there's a heterogeneity in their maturation status. But once they get into the tumor tissue, somehow this tissue actually program this neutrophil to become more like homogeneous, so that they will carry out the similar functions. So they will acquire similar functions within these microenvironments. So from here, actually, we would like to propose a working models. In the past, we know that from Andreas' lab, what he showed that under the resting state, mature neutrophil can go to the tissue and acquire certain tissue functions. And also there are various studies also show that under the stress conditions, the neutrophil development trajectory is still preserved. However, there is this transcription factor regulations when the cell moves from one compartment to another compartment. But then this actually raises the questions: if all of these cells that get to the effector site or the local site, each of them have a distinct functions represented by these different colors, how would this cell carry out a productive effector function? So this doesn't actually kind of make sense. So instead, we would like to propose that how this work is that, I'll skip that one. How this works is actually there is a uncoupling of this maturation program under the inflammations or tumor or any stress conditions. When the neutrophil there's an increased production of neutrophil, but then there's an uncoupling of this maturation program with this tissue differentiation program, in which it doesn't matter whether these cells are the mature neutrophil or immature neutrophil, they will then acquire this tissue differentiation program and acquire this function to carry out their function at the local site. So to summarize and to provide some perspective, we show that I hope that I convince you guys that the T1 immature neutrophil and mature neutrophil that we observe in the tumor can further differentiate into T3 neutrophil, and most likely they are pro-tumoral based on all our analysis, and currently we're doing various functional study to show that. And more importantly, we also show that there is this uncoupling of this maturation program and tissue differentiation program when neutrophils are activated and when they are recruited during inflammations. And this will then allow the neutrophil to adapt to the local microenvironment to carry out their functions. And the other point that I also want to point out is that immature neutrophil in this case can also be recruited as a reserve cell to the local site to ensure there will be sufficient cell to perform their effective functions. While this is good when you are generating a protective response, because in a way that these features will allow neutrophil to kind of increase their repertoire in their functions. However, when it's hijacked by the tumor, then this is actually not good, because then this will be help the tumor to grow and preserve the tumor. So finally, I just want to thank all the people in my lab, and to me, in a way that perhaps this will be the last talk that I'll be using kind of an A-star slide to show all my data and to give talk. And this group of people that I'm really grateful and thankful for the wonderful 13 years they have given me. Some of them are still in the lab. Some of them is already move on to do some other things. And I also want to thank all the SIGN collaborators for specifically for Ron for all his help and friendship, and also Anis and Evan for their help and friendship also. So and with that, I also want to thank my international collaborator Andreas and Renato. They are my partner in crimes. We always discuss many different things about neutrophil. So lots of inspirations. Our project is coming from discussion with them, and obviously other collaborators here. And as Matteo mentions, I'll be moved kind of in transition. So I'll be starting my new lab soon, or kind of started in some way already. In the Shanghai Immunotherapy Institute, led by Professor Chen Dong, and we are recruiting. If you're interested to do some neutrophil work, you can email me. So so thank you very much.
Thank you very much, Lai Guan, for a great talk and a fascinating journey through neutrophil development and function in steady state and within tumors. So, as you know, the questions to Lai Guan can be asked via Twitter. You should search for the account Global IMMUNOTALKs, find the tweet that says "Ask questions for Dr. Lai Guan Ang" here, and you can reply to that tweet with your question and mention the hashtag Global Immuno, and Lai Guan will answer the questions via his Twitter handle. So thanks again, Lai Guan. This was great, and thanks everyone for attending.
Thank you.
All right, bye-bye.